Thank you everyone for coming to this presentation. I'm going to present a new system that's used to measure the quantitative absolute elasticity using steady-state shear wave imaging. I'm actually going to present a specific implementation for use in measuring in vivo prostate tissue. So just to outline what I'm going to go through, I'm first going to describe what steady-state shear wave imaging is for anyone who's not familiar with it. Then go through the design that we have, the hardware design. Then I'll describe some of the processing that goes specifically for this system before I go through some validations with the Phantom and some clinical studies that we've been able to perform using this system. So what is steady-state shear wave imaging? Basically, it's something similar to MRE techniques where we generate mechanical wave inside the tissue using continual multi-frequency excitation. And once the wave is generated, we can then measure the elasticity by actually measuring the wavelength of the shear wave inside the phantom. This is as opposed to transient methods where you measure the speed of the wave. Instead, we're measuring the wavelength, but still to compute the absolute elasticity. So as a background for previous work that we've done, Ali Baghani has done a 3D system. He used color flow Doppler sequencing to track the tissue and provide a high enough frame rate. Separately, Hani Eskandari, along with other people in our lab, have developed a Bandpass sampling technique for elastography. So in this work, we kind of look to combine the two of them to provide 3D elasticity and extend that to use it instead of, like for Ali's work was with the ultrasonics research interface. But in this case, we want to extend it to use a more high quality conventional clinical ultrasound machine. So this is our prostate elastography system, and going through one by one, I'll start with the ultrasound machine. So we use the standard clinical EK Medical ProFocus ultrasound machine with the trans-rectal thrust probe, 8848, a sagittal array of that. We use a frame grabber to stream the data into a PC, and we get beam-formed IQ data. So this is the natural frame rate. It's not, regular frame rate. In order to get the 3D volume, we use this is a regular brachytherapy setup, but we modify it to have like a robot mechanism that sweeps the cradle to be able to get us a 90-degree volume. So we get a full volume of absolute elasticity by sweeping 100 planes with at 0.9 degree increments. In order to get the excitation through into the tissue to generate the shear wave, we use a transperineal shaker, which is a voice-called speaker type mechanism, to generate multi-frequency transperineal excitation. We use this Civco flexible arm that can be used to maneuver the transperineal shaker and provide adequate contact with the tissue to generate the wave inside the tissue or phantom. So how we do the processing is once we get the data through the frame grabber, we can acquire a bunch of frames, do correlation tracking, which was also developed in our lab by Reza Zahiri, and basically track tissue patches at a given plane. So for each of those tissue patches, we can get the oscillation at every for a given frequency. Since we know the frequency, we can just store the amplitude and phase values at each of those pixels. So in the end, for each frequency, we can get a complex phasor plane and do this for each of the frequencies. So just to note, when we do the tracking, the multi-frequency excitation that we use is on the order of 100 to 300. Well, not even limited to 300, but it's much higher than the sampling rate that we get off the BK machine, which is around 42 hertz. So we are well below the Nyquist rate. But because we're only looking for the phase and the amplitude of the signal, if we have a frequency, let's say, of fs, we can pick a frequency, and as long as what happens is it gets aliased down into the baseband, and as long as that single frequency doesn't fall on a multiple of the sampling frequency or half the sampling frequency, we can actually reconstruct at least the amplitude and the phase. So when we go to multiple frequencies, we can still do the same thing as long as when they come down into the baseband, there is no overlap. So then we can distinguish the signals and simultaneously process multiple frequencies. So then the idea is basically we do the same thing at all the planes that we've done to get a complex phasor image. And from there, we can then look at the shear wave direction, in whatever direction it is, in the 3D direction, compute the wavelength using local frequency estimation. So once we have the algorithm working, the first thing we did was to validate the elasticity measurements. And we used the CIRS 66 prostate phantom, which has hard inclusions inside a simulated prostate. And from literature, we have that the prostate is supposed to be around 15.9 to 15.9 kilopascals. And the documentation says the inclusions are 3 times harder. So we have approximate values of stiffness that we need to match our elasticity measurements to. So before I go into the actual results, this is an example of a phasor image that you can get. This is actually the absolute value of the complex phasor at a given plane. In this case, for the phantom, if I go back, we used multi-frequency excitation, so 144, 165, and 181 hertz at the same time. And these frequencies, again, were picked so that we can identify them in the baseband. So this is an example of the phasor images that you can get for each of the frequencies at a given plane. Once this is now 2 different planes with actual elasticity results, the 2D maps. We do compute this elasticity for all the entire volume, so we get it in 3D. And in this case, I picked 2 planes that have the inclusions in them, and you can see the scale in the middle is in kilopascals from 0 to 50. So our measurements do match up with expected values. So once we validated the elasticity measurements, we could then go to clinical studies and basically see if we can correlate our absolute stiffness values to pathology or to cancerous tissue. And we did 2 separate, we actually are still doing 2 separate studies, a prostatectomy study and a focal therapy study. I'll go into them in more detail individually. So with the prostatectomy study, we have patients who undergo radical prostatectomy at the Vancouver General Hospital. We obtained the pathology slides, which are marked with cancer and cancerous and non-cancerous regions. And then by registering them to the ultrasound, we can compare our elasticity volumes and slice them in the same way that the pathologies are sliced to compare the 2. So I'm just going to present a few examples. This is 1 patient, 2 different slides. On the far left is pathology slice, middle is B mode, right is our elasticity image. And you can see just visually there are correlations between the places where are identified with positively cancerous and our stiffness maps. This is another example for different patients, but again, 2 different slices. With this study, we actually did a full classifier-based machine learning analysis but using both the absolute elasticity as well as the phasor imaging component, phasor data. And with that data, we were able to get an AUC of 0.82 with regards to prostate cancer identification in the peripheral zone. The next study that's still going on this is a much newer study, but it's part of an investigation into focal therapy. And so the way we do this is they have at the BC Cancer Agency, you have whole gland sampling biopsies where they take 30 to 40 cores of the prostate rather than the conventional 6 that they take almost blind in regular biopsies. But in this way we can determine the size and location of tumors within the prostate. So then we have like a map of where they are. Again, we do multi-frequency excitation. In this case, we have a slightly lower range, 69, 75, 80. Again, they're spread out accordingly inside the baseband. But it's lower frequency because in the patient we found the penetration needs to be deeper to get into the, I think with the pubic bones and every structure, it allows us better wave generation. And so again, this is almost a similar imaging where you got on the left, this is now biopsy reports. The blue circles are negative cause. It's actually pink, but I've outlined again in green so you can see the positive cause. The middle is B mode and the right is our absolute elasticity maps. You can see that the shape of the image is different, the scan conversion, and that's because our absolute volumes are 2D sagittal planes that are swept 90 degrees, whereas the biopsy is those images are taken from the transverse plane. So this is interpolated into a 3D grid and shown as the transverse slice. That's why they look slightly different in terms of the scan conversion. So this is an example of one where, again, 2 slices in the same patient, and we have positively identified cancerous cause, and our elasticity maps show visual correlations between the two. And this is another example of another patient. With again, similarly, we have some visual correlation between cancer and our stiffness maps. With this study is much newer. We haven't yet done. In future work, we'd do more of an analysis, like a classifier based, like we did with the prostatectomy study, to investigate correlations with cancer. So to conclude, we've developed a generic ultrasound absolute elasticity system. We've used the clinical BK machine to do that and investigate the correlation between elasticity and cancer. I've demonstrated numerical and visual correlations between tissue stiffness and cancerous tissue. And because of allowing the generic use with the Bandpass sampling and the 3D motor rather than 3D probe, it's a generic platform that can be applied to other organs, let's say liver or kidney, which is also being done in our lab. But I presented the specific application of the prostate in here because we have a few studies in there. And as I said, we'll probably do further analysis with the folk therapy study. Thank you.